Sorry about that. I didn't unmute. Thank you, Pride, for the message. Welcome everybody to a session of Intellectus Campus. I'll proceed with the introductions. So, Mr. Newten Handireketi is the Deputy Director of Health Research at Zimbabwe's Ministry of Health and Childcare. He boasts over 2 decades of experience elevating healthcare systems in the nation. He coordinates research endeavours with a focus on health system strengthening, adeptly managing work plans, and fostering collaboration. Newten oversees performance assessments, ensuring research efficiency, and maintains a robust project database. Newten was previously a research scientist at the National Microbiology Reference Lab. He consolidated expertise, and additionally, he remains pivotal in shaping public health research in Zimbabwe, driving its progress through collaborative agreements and impactful initiatives, and cementing his status as a stalwart in the field. We have pre-recorded the session. Dr. Hector Carvallo is currently between Paraguay and Buenos Aires. He is a well-accomplished physician with expertise in endocrinology and is certified by the College of Physicians of Buenos Aires, and his specialty is in infectious disease. Unfortunately, one of the cabin crew members won't let him use his phone, and his plane's delayed. So we hope that he lands and is allowed to use his phone shortly. I'm going to flick through just a couple of reminder slides. And I'd just like to remind everybody of the chain of infection. So tonight we will be discussing the infectious agent, which is the malaria parasite, the reservoirs, which are humans and asymptomatic carriers and other mammals. The portal of exit is the mosquito sucking the blood out of the carrier. The means of transmission is the vector, which is the Anopheles mosquito. The portal of entry is the bite of the mosquito, and we will discuss susceptibility of the host, but we are going to focus quite a lot tonight on the vector. I just remind you that we encourage constructive controversy. There is no one way. Newten and I disagreed within the recording on one particular point, but I'm coming at it as a clinician and he's coming at it as a researcher. And that's the way that we actually achieve consensus, is we debate and we come up with a conclusion. We also are emphasizing African solutions for African problems. A lot of people in Europe and the US live inside and they don't realize that most Africans spend their evenings outside on a veranda, and we are putting the nets and the barriers between the outside and the inside, and we as humans sit outside. And that was a lesson I learned from today. Today you will get to know your enemy no matter what it is, and it's critical in infectious disease. And as a lesson, the only way we can beat this is as a team, and to stay ahead of it. So welcome everybody to another session, and Pride will start to play the recording that we did earlier. And at the end of it, I'll open the floor to questions and answers. So thank you, everyone. And Pride, please can we play the recording? So welcome, Newten. It's wonderful to have you here. And thank you for taking us through what's new in malaria. And I'll run the slides if you want. And just say next slide and I'll change. So tonight we're presenting on the approach to malaria and what is new. And we're very privileged to have the Deputy Director of the National Institute of Health Research in Zimbabwe with us tonight. So please go ahead and I'll progress with the slide.
Thank you very much, Dr. Stone. I really appreciate today I'll be representing myself in this presentation not the official position. I found it fine to share some new approaches to malaria, what is new in terms of our approach to malaria. Yes, you can. Okay, so this is gonna be my my presentation outline. I will look at the overview of malaria entomology, overview of malaria parasite or case control. Then I will have a look at community education. So let me also put a caveat to say that what might be new may not really be new to others, but we are looking at some new innovations which are coming on. But the people who are working on the new innovations, they already know a lot about what they are working on. But to some sectors who are looking at malaria, they might not be having a lot of information about those areas. So we will look at some areas, new emerging issues around malaria entomology, emerging issues in parasite or case control, as well as community education. So we are going to take it from the basics. We are going to start from the simplest issues around malaria, so that it is fine for us to just talk about the basics of malaria. So malaria is a disease that is caused by a parasite. The parasite is called Plasmodium. So Plasmodium, we have many species under Plasmodium, so we'll talk about those ones. Then this parasite is transmitted by a female Anopheles mosquito. So these are mosquitoes under the genus Anopheles. We have several species under the Anopheles genus. Let me take you through the basic malaria epidemiology. We are simply looking at the distribution of malaria epidemiology, control, prevention. So Anopheles, the Anopheles mosquito, borne infectious disease, that is malaria, has resulted in about 249 million cases globally. And out of the 249 million cases, 233 million, that is about 94%, were in the WHO African region in 2022. I couldn't get the most recent approximations of 2023, but I think the distribution in 2022 is recent enough for us to actually see that the largest burden of malaria is in Africa, and approximately 608,000 deaths occurred globally due to malaria in 2022. So that is a big number. And globally, estimated 2.1 billion cases and 11.7 million deaths were averted between 2000 and 2022. So we can actually see the effect that malaria could have caused because of the new innovations which were on board and some old methods which have been used to prevent these deaths. So if it was not because of these interventions, we would have lost quite a huge number of people, about 11.7 million people could have been lost. So most of the cases, about 82%, and the deaths averted were in the WHO African region. Let me also go still on the basics. If we cannot understand malaria, we can't really control it. So we are going to look at several issues around malaria, why it becomes very difficult to control malaria if we are not understanding it. I will try to explain a number of issues why we are having challenges in controlling malaria. But before we get there, let me go to some basic definitions that we need to know when we are talking about malaria. The first one relates mainly to the vectors, that is bionomics. When referring to mosquitoes, this term translates to the biological, ecological, and behavioral characteristics of a species or population. That is it. I'm going to elaborate on this one because we are going to meet the same term again in this presentation. Then the sporozoid, this is the life stage of the malaria parasite in the mosquito that is capable of producing infection in humans. So when a mosquito bites, it releases these sporozoids into the human being, and the sporozoids will then proceed through the life cycle of the Plasmodium parasite. I will give a slide where I will just show the Plasmodium parasite life cycle. Then we also have this circumsporozoid stage specific protein on the outer surface of the sporozoid. So this protein can be used for testing to detect the Plasmodium parasite.
So Newton, I'm obviously much older than you, and we've had this discussion before with my ability to run technology. But when I was a student, we used to do thick and thin smears and Thea ring.
Yeah.
Is that the circumsporozoid?
Not really, because we can't see a specific protein under a microscope. But what we can see is actually the parasite itself inside an erythrocyte. But I think we'll talk a little bit more about the diagnostics, yes. But thank you very much for that. Then another basic definition is entomology. Entomology, that's the study of insects. In this case, we are mainly interested in the mosquitoes and their relationship to humans, the environment, and other organisms. So we'll talk about what is new in malaria entomology. Then I also thought that there is this other term which is malaria epidemiology. That is the study of the determinants, distribution, and patterns of malaria in populations, and how it is applied for control and prevention of the disease. So that is malaria epidemiology. So I'm sure quite a number of you have come across this nice picture demonstrating the life cycle of the Plasmodium parasite. So some catchy things to take note of here is we have the mosquito stages of the parasite. We have the stage where the parasite is in human liver. We have got the human blood stages. So the life cycle of the Plasmodium parasite has been studied quite extensively. As you can see that the mosquito, just for starters, we have the mosquito there at number 1, taking a blood meal. Then as it is taking blood meal, it releases the sporozoids that we talked about. They are released into the human body. They get to the liver. In the liver cells are infected, and the parasites will multiply to form what are called schizones. The schizones will rupture the hepatocytes. These are the liver cells. Then these released parasites will infect red blood cells. This now brings us to the question from Dr. Jackie. So this is the ring form of Plasmodium that we can see under a microscope, where we were saying that this is not the circumsporozoid. The circumsporozoid is actually a protein found on the Plasmodium parasite. Now, so some of these will then mature to form the male and female gametocytes. But some of them will still go through the same cycle where they can multiply and increase in number. But as they are multiplying, they can result in rupturing of the red blood cells, leading to anemia in patients who are infected with malaria. And when a mosquito is now taking a blood meal, it also ingests the male and female gametocytes. Then these ones will fuse in the mosquito, then injected back into the next victim. So this is how extensive the malaria a parasite life cycle has been studied. But the question is, if we understand this life cycle of the Plasmodium parasite so much, if we have got such a thorough understanding of malaria parasite, how come that we have so many cases of malaria that are going on? We have a huge challenge here. And this will take us to the next slide. So if we understand the life cycle of the Plasmodium parasite so much, why is it difficult to eliminate malaria? So actually the complex life cycle that we have seen, that the parasite is has got two hosts, the human as well as the malaria, the mosquito. Then in the human, it gets into the liver cells, then it gets into the red blood cells. That complex life cycle makes it difficult to eliminate malaria because we need to know where to focus so that we can target stopping the life cycle. Then we also have another challenge of parasite drug resistance. As we are treating the parasites, they are also trying to adapt and evade the effectiveness of the medicines which are used to treat them. Then with the current vaccines which are available, we have challenges with their efficacy as well as duration. You know, when you are infected with the pathogen, after some time, you can still be infected again. The same as with the vaccines. You receive the vaccine, after some time, the protection can only go for a certain duration, after which you can then be reinfected. So you don't get a lifelong protection from malaria. So that becomes a challenge. Then we have a challenge because of malaria endemicity. There are some areas where we have got high transmission rates, that is in tropical regions as well as tropical regions. Where we have got high transmission rates and we are trying to eliminate malaria, the moment we are trying to come up with interventions, we treat patients, they go back into the community, they get infected again, and the cycle continues. Then we also have another challenge on poor surveillance and monitoring cases or hotspots of malaria. This becomes a challenge because we might not know the real burden in the community or some hotspots. So there will be ongoing transmission and it becomes difficult to eliminate malaria. Then going to the vectors, there is vector resistance to insecticides. We'll talk more about this one. As we are trying to control malaria, focusing on the mosquitoes. The mosquitoes are the mosquitoes are controlled by insecticides, but the mosquitoes themselves are trying to resist the effectiveness of the insecticides. Then there are changes to other vector bionomics. Remember, we talked by bionomics, we talked about the mosquito biology, the mosquito behavior. So I'm talking about other vector bionomics because resistance to insecticides is also part of bionomics. But the other areas which complicate elimination of malaria are changes in the vector bionomics, such as anthropophily. Anthropophily, that is their desire to feed on human blood. Then, so because the female mosquitoes require human blood, which has got proteins that enable them to lay their eggs. Then there are some mosquitoes. So it's actually an area, I'm sure if we have got some entomologists on this call, they understand, they study the mosquito behavior. So there are some mosquitoes which like to reside outside of homes. What it means is that if you've got some interventions where we are trying to prevent getting bitten by these mosquitoes inside homes, as soon as you go outside and transmit malaria. Then we've got some mosquitoes which are endophilic. These ones like to reside inside homes. Examples are Anopheles funestas. So if it is residing inside the homes, then you can actually come up with interventions for those ones, like spraying the homes and using some nets. But spraying inside homes and using nets will not be effective against those ones which are exophilic. Then we also need to study a mosquito behavior to see which species are exophagic. These ones usually like to feed outside the homes. So as soon as you go outside, they've got a tendency to get attracted to you, and they want to have a blood meal on you when you're outside. Then there are some mosquitoes which have got a behavior of just wanting to feed inside houses. So there are some innovations now. I think for entomologists, it is work which they've always been doing, doing some surveillance, looking at this vector bionomics and characterizing the mosquitoes. But this is an area, quite an interesting area for entomologists. We are still on why it is difficult to eliminate malaria. As you can see that the list is very long. It shows why malaria has been a persistent problem over a number of years. And I've just given you under malaria epidemiology, those figures, those are alarming figures. One of the reasons which make it difficult to eliminate malaria is human behavior. You give them mosquito-treated nets, they will not sleep under those nets. They use those nets for fishing. So it's human behavior. And what then happens? The insecticide gets washed when they are doing fishing, and they don't want to sleep under those nets. They get bitten by mosquitoes, and transmission will still continue. Some people, you spray their homes, then when it is about to be a festive season, they just paint their walls, not knowing that they are already interfering with some interventions which have already been put in place. So in some rural areas, they can also rub the surfaces with soil, which try to beautify their homes. So this is human behavior, which makes it difficult to eliminate malaria, because in the end, these people will not be protected from malaria. Then we have got this challenge of asymptomatic cases. These become reservoirs of infection. So we need some new innovations to detect malaria even in asymptomatic cases. And these asymptomatic cases are usually very difficult to pick through passive surveillance. An asymptomatic case will not present to a healthcare facility to get treatment. So they will continue. But if a mosquito bites an asymptomatic case, they can say the mosquito can still proceed to transmit the infection to another person. It becomes one difficulty in eliminating malaria. Then we've got challenges with cross-border transmission. In Zimbabwe, we have got many cases of malaria along the border districts. You look at Mutasa, Chiredzi, going up Nyanga, going further up to Mudzi district. We have centenary districts. You will see you will see that we have got a high number of malaria cases. It is because of cross-border coordination, which may need to be strengthened, without which it will be very difficult to transmit, to control and eliminate malaria. And I think that is not a problem which is unique to Zimbabwe. In irregular patterns of rainfall, we've got some areas which traditionally have not been used to receiving high amounts of rainfall. Then the world is becoming warmer and warmer, and the vectors will find it some areas which did not used to be habitable, more habitable, and they'll be able to transmit infection. So it becomes a challenge now because the boundaries are being pushed out for the vectors to transmit infection. Then we've got travel and migration. It becomes one other challenge to eliminate malaria because someone can be an asymptomatic case of malaria, then we simply travel to another area where malaria had been eliminated. There are mosquitoes there which are vectors. They can bite that individual and start the transmission. Everything else will then proceed. Then we also have a challenge in terms of commitment. So commitment can be political commitment. We need commitment from our leaders as well as financial commitment. So political commitment, this is where our leaders will take some, will come up with some declarations and targets to make sure that we are going to eliminate malaria. Then they can also mobilize financial resources to make sure that there is elimination of malaria. Then the huge elephant in the room is health systems issues. So according to WHO, health systems has got several pillars. So within each pillar will contribute in a specific way to elimination of malaria. I'll talk about this one towards the end of this presentation.
So Newten, I just want to ask you because I want this to be interactive.
Yeah.
So if we look at your slide, I'm going to Teresa next week. I've already made a phone call to say have they got malaria around, and they've said yes, it's still around because of the recent rain, which is unusual in May, which makes your point. I will sleep under a mosquito net, but I will be going outside at night with a spotlight, and I will be sitting in an open area and get bitten. So actually the mosquito net's a total waste of time because I'm intending to expose myself at night with lights in an outdoor area where I'm very likely to be bitten. And when I phoned through for the local knowledge, they said just make sure you've got some Lumartin, and if you get sick afterwards, because they've got resistant malaria down there, if you get sick afterwards, just take a three-day course of Lumartin. So having listened to you so far, I'm actually contributing to the problem. Because I'm going to go down there, I'm going to get bitten outside. If I get malaria, I will be able to transmit it for a few days before I become symptomatic. Because I think it is, if I'm not mistaken, falciparum is a 12-hour cycle, and it's 16. One parasite turns into 16, which then turns into 16 times 16, and you get that exponential rise in parasite count. So the behavior of what most Zimbabweans do when they go on holiday is exactly what's contributing to the transmission of malaria.
Thank you very much for that, Doc. Yes, you are quite right there. And Chirezi is an area where we have been having a lot of activities in terms of malaria control. It is because of the sugar plantations. There is a lot of irrigation, there is a lot of water, which becomes very good breeding sites for the mosquitoes. As the mosquitoes are increasing in numbers, we also have high chances of someone getting bitten by those mosquitoes. They are also implementing very good measures there to eliminate to control the vectors themselves. But thank you very much for that contribution. So human behavior it will contribute quite a lot to transmission of malaria. So having said this, so this list is not exhaustive as such. We can have more and more reasons why it is becoming difficult to eliminate malaria. But I don't want to put out the spirit of fighting malaria and eliminate it. We still have some hope. And our hope, we have got malaria control, what is in place and what is new. So traditionally, malaria is comprised of, malaria control is comprised of these basic three components. We have to control the vector. We need early detection and effective case management. So this is where we are dealing with the parasite itself. And this is the lovely choice for malaria control programs. Then we've got community education. But the issue here is we need to avoid working in silos. So that's why I said issues around health systems will then kick into place, where we need to find how we can coordinate vector control, early detection and effective case management, as well as community education. So for starters, the malaria vectors, let's go to vector control. For starters, malaria vectors, we have got different vectors that can contribute to transmission of malaria. We've got Anopheles gambier, then we've got Anopheles arabiensis, Anopheles coluzi, Anopheles funestas, and Anopheles stephensii. In Zimbabwe, we have Anopheles arabiensis, Anopheles gambier, Anopheles funestas. Anopheles funestas, this one is a very efficient vector in terms of malaria transmission. So what is involved in malaria vector control? These are measures to reduce the contact between mosquitoes and humans and to reduce the number of mosquitoes that reach the stage where they can transmit the sporozoites, the parasites. Remember, we're saying that when a mosquito is having blood meal, it will release the parasites into the body of the target, and the parasites will then go to the liver, the red blood cells, and the cycle continues. Adult vector control. So in terms of vector control, we can control the adults, we can control the larval stages. So in terms of controlling the vector, the adult mosquitoes, this involves reduction of human mosquito contact by insecticide-treated nets. So we just distribute insecticide-treated nets to individuals so that they sleep under those insecticide-treated nets. Then we also have indoor residual spraying. So this involves spraying of walls, sprayings of houses with the insecticides. Then people can also use mosquito repellents, protective clothing, just to make sure that you avoid being bitten by the mosquitoes. I think, Dr. Jackie, this is a question that you're asking, something that you're contributing to. Then screening of houses. So screening of houses, we are saying that we don't want the mosquitoes to get into the houses. Those ones, the endophagic, the endophilic, those ones which like to reside in homes, those ones which like to, which have a behavior of a feeding inside homes, we don't want them to get inside. So we can put some screens on our windows. I'm sure for those who have traveled, you have seen in some certain areas where there are some screens on the windows, so that even if you open the window, the mosquitoes will find it difficult to get into the house. So it is to avoid. So this is the screening of the houses and other housing improvements. So the design issues will become very important if it is in malaria endemic areas. As we are designing our houses, we need to make sure that we take cognizance of issues around malaria control.
Just one thing though, we've been talking about African solutions for African problems. So since I was a child, I've lived with mosquito screens, but then we all go and sit on the veranda with the light on outside. So one of the things we really should be aware of, I mean, next week we are taking someone who's here from Australia to go and look at rhino and game. So we're going to be outdoors. My concern is when that person goes back to Australia, they won't recognize why he's got a fever and he's unwell. So he will go back with malaria. But in actual fact, I would say the vast majority of the patients I have seen with malaria have been on holiday to a malaria area. They're on a houseboat, or they are camping, or they are doing an outdoor activity. So putting screens on houses is not going to protect people if they socialize outdoors. And it's a message, probably my message from this is I'm taking long sleeved shirts and mosquito repellent to put on myself, because I know I won't spend my time indoors next week. So whether there's mosquito screens on or not, I'm going to get bitten.
Thank you very much for that contribution. So you are bringing a very important perspective that malaria control is not a one so we say avoid working in silos, intervention that works. For example, insecticide treated nets are not just provided in isolation. In many cases, they can be provided with a combination of indoor residual spraying, as well as taking some mosquito repellents. Someone can also take malaria prophylaxis. So it's a cocktail of interventions which need to be taken at the same time in order to control the larval stages. So it's called larval source management. So we know the life cycle of the malaria vector. It involves a breeding in water where they can lay their eggs. Now, if we remove that water, the mosquitoes will not have anywhere to lay their eggs, and we can control the mosquitoes that way. So source reduction by, we need to reduce the source of the larval stages by larviciding. This is just spraying of water with chemicals that can kill the mosquito larvae. Then intermittent irrigation, spacing, then biological control. We can actually use like fish that can eat the mosquito larvae. Then we've got environmental sanitation, water management, and drainage. So when we're talking about malaria, in many cases, people look at the healthcare system, but to control malaria, it's not only the healthcare system, but it also involves a lot of other stakeholders. For example, our colleagues from the environment, our colleagues from finance, our colleagues from who design buildings. So these are some areas which need to be looked at when we want to make sure that we can control malaria. So from vector control, let me go to early detection and effective case management. So in order to control malaria, we need prompt diagnosis. So the traditional diagnostic methods have been rapid diagnostic tests, or microscopy, or they can be used in combination. So when someone has been confirmed, the clinicians, this will be now their area to assess severity of the malaria case, whether it's simple malaria or it's a complicated malaria. They may also need to make further investigations depending on how they are managing the patient, trying to understand some underlying conditions which will guide their decision in terms of treatment. I will get back to this one a little bit later. Let me go on to the treatment. The clinicians then come up with treatment plans where they can use artemisinin combination therapies, in taking cognizance of the age, weight, severity of infection, whether it's now complicated malaria, in which case they may need to consider injectable artesunate for complicated cases, which is replacing quinine. Then considering primaquine, which can also be effective against the gametocytes. So primaquine is gametocidal. So this will be a decision that will come from the clinician, who is handling a particular case. Then apart from treating malaria, there might be need for some supportive care in terms of providing relevant fluids, providing some rest to the patient, monitoring experts in malaria case management and treatment of malaria. They can go very far explaining these areas. Then such as temperature, potential complications, monitoring parasitemia, which can also be a marker to resistance of the parasite to the available medication. if there's need for referral of some severe or complicated cases, they'll make the decision. Then we also have some innovations in terms of digital health technologies. For example, someone goes home, continues to take some medication. There might be need to have further communications with the clinicians. This is where telemedicine solutions will need to kick in, innovations in mobile applications, then electronic health records for patient management and surveillance will then kick in.
So, sorry to interrupt you again, Newton, but one of the things I think is very important as a message to go out, particularly to junior doctors, is that malaria is actually a clinical diagnosis. And a negative test does not mean that the patient is negative for malaria. It means you need to repeat the test with the next cycle of fever. And just as somebody who's had malaria, because you know when you've got malaria, the rigors, the headache, the vomiting, it's hard to mimic. But as someone who's treated people with malaria, I've had people come in with dark urine because of the hemolysis, with anemia, and they say, I've had a negative malaria test, where in actual fact, they've got a classic history. 10 days ago, they were in a malaria area, they've got fever, they've got headache, they've got no other cause, like a sore throat, for the fever and the headache. They've got rigors. When the parasite isn't causing rupture of the cells, they're fine. And they become extremely unwell for several hours, and then they're fine again. When you've got that history, I would advise people that if the urine is becoming dark, and particularly if you can get a hemoglobin which is low, and if the platelets are low, then you need to proceed with empirical treatment. It can be diagnosed clinically. Would you agree with that? That if you can't get a test, don't delay treatment.
Okay. So I think this is something that may need to be well thought about, because currently the guidelines are saying that for malaria treatment, you need a positive diagnostic test. It can be an RDT or microscopy. And let me also indicate that microscopy also needs to be in good hands, so that we don't have, so that we don't have false negative results for malaria diagnosis. Then RDTs, again, there are some quality assurance issues which come into play. So, but I'm sure the guys from the lab know this area very well. So there's also need to just communicate with the guys who do the diagnostics, just to make sure that everything is in order in terms of diagnostics. But thank you very much for bringing that perspective. Let me also proceed with early detection and effective case management. Like I indicated, that the current guidelines indicate that all presumptive malaria cases should be confirmed by parasite detection methods before treatment, as follows. We can do microscopy, a thick or a thin smear. We can use our RDTs, which are point of care on the bedside. So these RDTs can detect a histidine-rich protein. It's a part of the protein on Plasmodium falciparum. And we can also detect parasite-specific lactate dehydrogenase. And we can also detect Plasmodium aldoles from the parasite glycolytic pathway found in all the Plasmodium species. Now, we have methods which are more sensitive. The polymerase chain reaction. We also have new innovations. There might be new in some areas, but a technique which has been there, that is a nucleic acid sequence-based amplification. That is the NASPA technology. So we have malaria parasite capillary electrophoresis. So this one involves using thin capillary tubes to separate parasite DNA or proteins based on size and charge. So this can be used to detect drug resistance, then to also quantify parasitemia, for clinicians, they may want to know from the before treatment, what was the parasitemia? After treatment, what is the parasitemia? It becomes handy. Then speciation, now to be able to identify whether it's Plasmodium falciparum, Plasmodium vivax, Plasmodium ovale, Plasmodium malariae, the different Plasmodium species can be detected by using this method. So the advantage is that these molecular methods, they have high sensitivity and specificity. Then analysis can take from minutes to hours. Then a small volume of sample can be used. Then we can also take advantage of automation. We are still on early detection and effective case management. It's a very huge area that we cannot really exhaust everything. So now getting a little bit deeper, safe and effective radical treatment, typically with primaquine of Plasmodium vivax malaria, currently requires 2 diagnoses. The presence of Plasmodium vivax and glucose 6-phosphate dehydrogenase status. Why the glucose 6-phosphate dehydrogenase status? It is to take care of those patients who have got glucose 6-phosphate dehydrogenase deficiency condition. If primaquine is provided to those patients, that can result in hemolysis. So in as much as we want to do good by treating the malaria, we might actually do some harm. So for clinicians, these are the further investigations which may be of interest, and this is a recommendation from the WHO. And that 2-stage way of diagnosis in areas where we are suspecting presence of Plasmodium vivax will be necessary because if it is Plasmodium vivax, you may want to consider primaquine that is also gametocidal. Otherwise, we may have some relapse. So alternatives to primaquine may be considered for chloroquine, but chloroquine may not eliminate the hapnozoids. Then artemisinin combination therapies may not also eliminate the hapnozoids. Then we have tafenoquine with a trade name of Cosenix. This one is approved for patients with G6PD deficiency. And then hydroxychloroquine, then atovac and proguanil, but this one may not be recommended as the first-line treatment. So this will be some points of interest for further discussions. So to avoid irrational use of artemisinin-based combination therapies, the first-line treatment for uncomplicated malaria and reduce the drug pressure on parasites and could contribute to more effective treatment of other or to more effective treatment of other cases. So we talked about vector control, we talked about early detection and case management. Another area is malaria, is community education. There are lots of areas which have been focused on, including, so this is the empowerment of the communities to identify and manage malaria. By understanding the signs and symptoms of malaria, we need the communities to understand those and its transmission so that we can demystify malaria transmission. Importance of insecticide-treated nets and IRIs in prevention. I said that in some communities, you can actually provide them with the nets, and they are not used for the purpose. For drug adherence in the communities, there are some who say, I'm not feeling okay, there's no need for me to finish treatment. So we need to educate them on treatment condition. Then eliminating breeding sites, they need to know that they don't need some breeding sites around their homes and level control. Then we need to take care of malaria misinformation. So some new approaches have been We found they are emerging in terms of vector control, on new insecticides, in terms of diagnostics, new medicines to treat infection, combination therapies, the vaccines, sterile insect technique, transgenic mosquitoes. We have got some surveys like the malaria therapeutic efficacy studies. Then we have got malaria indicator surveys. And then we need a systems approach to innovations where we adopt a systems approach to eliminating malaria. So I'll quickly go through the innovations. So in terms of new insecticides, over 2 billion insecticide-treated nets are distributed globally to prevent malaria. So they've been distributed since 2000, and they are treated with pyrethroids only. So mosquitoes in many areas are now, the challenge is that mosquitoes in many areas are now resistant to the pyrethroids themselves due to knockdown resistance mutations in the mosquitoes. So there are new insecticides, for example, pyroclostrobin, which can be a vector, which can affect the vector nervous system. So it can be used in areas where we are having these mutations. Then we have got nanoparticles, which can be, which is use of nanoparticles, very small particles to deliver insecticides and leaving the environment friendly. Then we've got botanical insecticides apart from pyrethrum. So pyrethrum is a chemical derived from chrysanthemum plant. So we also have neem oil, which is being considered for use in areas where we are having these mutations. So these are some of the new innovations. In terms of diagnostics, there is more emphasis of nucleic acid amplification technologies for detection of low-density malaria infections. They can detect as low as 1 parasite per microliter. They are highly sensitive because if they detect 1 parasite, they can amplify, make many copies of the single parasite to make them detectable. Then these nucleic acid amplification technologies can be qualitative, where we just want to know presence or absence of the parasite. Or they can be quantitative, where we now want to quantify how many parasites are there per microliter, for example. So these methods can also involve genotyping. Genotyping is an advantage for distinguishing recrudescence from reinfection. Then detection of drug resistance mutation will be another additional advantage of using nucleic acid amplification technologies. But at the moment, nucleic acid amplification technologies are currently considered for research purposes only. They are not used for routine care because of their cost, complexity, human resources needed to perform these ones. So we've got different types of these nucleic acid amplification technologies for malaria detection, including PCR. The PCR can be nested PCR, quantitative PCR, it can be real-time, reverse transcriptase PCR. So these are different forms of PCR. Then we also have another nucleic acid technology, which is loop mediated isothermal amplification. Then we have quantitative nucleic acid sequence-based quantitative nucleic acid sequence-based amplification. So these nucleic acid versions. So how do these ones differ? They differ in terms of the equipment, infrastructure, pre-amplification, sample processing that is required. They also differ in terms of reaction temperature. Some can, the reaction temperature can occur at a single temperature, but with process, time to result, complexity, limit of detection, but just giving an overview that we've got these solutions. The new medicines to treat infection, we've got some promising compounds such as carbapenem, codenamed KAE609, Janaplacide, KAF156, which are promising novel anti-malaria compounds in advanced stages of development to take care of some resistant forms of the parasites. Then combination therapies, we've got the vaccines, sterile techniques, transgenic mosquitoes that we talked about. Then coming to combination therapies, there are people who are already working on combining the current artemisinin with coumarin. We've got this group. So we've got other groups of researchers who are trying to come up with more combination therapies. Then we've got vaccination. We have these vaccines that we talked about, which are being done in Kenya, Ghana, Malawi. And current data has shown that these have been associated with reduction in severe cases and a 13% decrease in early childhood deaths. Then second malaria vaccine, this one was recommended by WHO recently. So this is something new in malaria. So was recommended by WHO in 2023 and is expected to increase broad-scale vaccine supply in high-patent areas. Then we have got this sterile insect technique. So this one relies on alternative mass release of non-biting sterile males, which mate with the monandrous wild females. So these monandrous wild females, they only mate with one male, and because it is sterile, there are no fertile eggs which are produced there, leading to elimination of the mosquitoes. Once mated females are permanently sterilized due to mating-induced refractoriness, which results in population suppression of the subsequent generation. Then we've got some transgenic mosquitoes. These are some innovations, vectors genetically modified to produce antiplasmodial compounds in their gut that blocks transmission of the parasites. So it may be antiplasmodial or just antimicrobial compounds which can be in their gut. So two antimicrobial compounds, for example, a gene from, so these mosquitoes can be genetically modified with a gene from secretions of the African cloud frog, Xenopas levis. Then another gene, the second gene, secretion, which results in secretions from the European honeybee, Apis mellifera. So the products of these genes will result in destroying the parasites. So the aim is to suppress or replace the wild-type malaria vector populations and reduce transmissions. Then now we've got some studies, therapeutic efficacy studies, which measure effectiveness of malaria treatment. Because we need to, for clinicians, they need to know whether the medicines they are prescribing are effective against malaria, which they plan to treat in the population. For example, if you know that, for example, in Zimbabwe, most of our cases are due to Plasmodium falciparum, you need to be confident enough that we still have Plasmodium falciparum in the population. And we've got merozoite-specific protein 1, merozoite-specific protein 2, group genotyping. This is done to differentiate Plasmodium falciparum recrudescens from reinfection. Then we have capillary electrophoresis that can be carried out during the surveys. Then we have got malaria indicator surveys, which can be repeated every 2 to 3 years. The purpose of this surveillance to determine malaria burden in defined populations, so that we can guide the clinicians' index of suspicion when someone presents with fever, nausea, vomiting. Then the indicators include malaria in the population. This will be an indicator of usage of the nets. So providing them does not translate to usage. IRS coverage, then health-seeking behavior for malaria treatment, malaria prevention and control interventions coverage. Then the one which I said, I'm now getting to the end of my presentation.
Right, can we end the call? I think it dropped out. So thank you, Newton. That was an excellent presentation. And I've got all the references on the slide. I am conscious of time. We've just hit 7:30. I'm just going to go through some of the clinicians' comments. And I think that Dr. Rapiti and Dr. Landman have both said that we don't have the luxury of time. And people have also asked questions regarding what we do when there's no access to testing. So I would say Dr. Landman has said, agreed, I understand the public health approach and the need to prevent resistance, but I will never underestimate the disease. I've had 2 patients over the years who woke up completely healthy and were dead by 10 p.m. that night from pulmonary malaria. So I'm a bit paranoid that once it's too late, there's nothing you can do. And I think this is why I'm encouraging the controversy, is that from a clinician's point of view, particularly with children, we need to not underestimate Plasmodium falciparum. It has a very rapid, it increases 16-fold with each cycle. And certainly I have seen patients who have had cerebral malaria who've never been the same since. And I recounted a case in North London where I walked to you earlier, Newton, where I walked into the hospital doing a locum on a Saturday morning. And there was a young 3-year-old boy who was in bed, who was a Black Nigerian. And he had a pyrexia of unknown origin, and he'd been there for 2 days, and every test they'd done on him was negative. And my first question, which was not, I mean, I was fairly new, I just landed, was, where did you spend Christmas? Because it was January and it was 1994. And his mom said, oh, we were in Lagos with his grandparents. And I said, well, what does the malaria smear show? And everybody looked at me as though I was crazy. And they said, oh, we didn't think of doing that. So, that child had a parasite count of 11% and was progressing towards confusion in a casualty, in a teaching hospital, and nobody thought to do the malaria screen. And he was commenced on medication before we got the result because the history was so clear. So I agree wholeheartedly with the clinicians, and that if you can't test, guidelines are called guidelines because they're guidelines. And we have to use our common sense when it comes to treating a potentially fatal disease. And Dr. Rapiti, thank you for your comment on that. Lydia, in Namibia, I agree with you 100%. What do we do when testing kits are out of stock? We rely on our clinical judgment, and we mustn't, as African trained doctors, underestimate our clinical acumen. We mustn't defer to guidelines that are often written by people who are outside of Zimbabwe. So I'm going to ask another question. Is the female Anopheles mosquito attracted to a particular blood type? Sometimes you can be in the same room with many people, but only a few individuals complain of getting mosquito bites. How are mosquitoes able to pick this up? And you enlightened me earlier regarding how some mosquitoes are attracted to blood at certain times of their life cycle. And can you answer that question? Just unmute your mic.
Thank you very much for that question. Can you hear me, Dr. Jake?
Can hear you clearly, yeah.
Yes. There is some association, of course, which has been noted in certain circumstances that mosquitoes tend to be attracted to certain individuals more than others. This is the complex behavior that I was talking about under bionomics. So I think more research will also be needed in this area to know whether someone is attracted, the mosquitoes are attracted more to someone with blood group A, B, or even other different blood group systems. I don't know if there's anyone else who has got more insight into this one. Thank you.
I'm not aware of any research into it. I don't know if anybody on the group can comment, but I do know that certain people are consistently more susceptible to mosquito bite, and they seem to be attracted to them. And I'm not sure what the reason is, but I would imagine if the mosquitoes can pick up blood at night and they're looking for blood, they obviously have a sensory system that we're not aware of. And I'd say if you are susceptible, certainly my lesson, and when I go away next week, I'll be using mosquito repellent in long sleeves when I'm outside. So, I think if the mosquito is able to pick up a repellent, it obviously is able to pick up something that's attracting it to certain people. And if you are one of those people, the advice would be to use DEET or another mosquito repellent. You mentioned the research on neem powder. I certainly have seen a lot of diabetics improve on neem, and I think that it's extremely bitter, which certainly in a lot of the cultures in our region is popular because it's then thought to be effective, because bitter medications generally are seen to be effective. So, citronella certainly I've seen work, and I think the message really is handle the victor. Would you agree, Newton?
Sorry, Jackie, you are breaking a bit.
I was just saying that if you are susceptible, it's a good idea to use what works. Neem powder you mentioned is being researched, neem oil, citronella oil. There are many natural remedies as well as the pyrethrins that actually repel mosquitoes.
Oh, yes. I think that will be a good idea. Like the question which was being asked on whether blood group can be a determining factor for someone to attract mosquitoes. It's not really one answer. It's not only one factor which can result in someone attracting mosquitoes or getting attracted to mosquitoes. It could be, of course, yes, there are some studies which have shown that blood group, the blood group is in some way associated with the mosquito bites, but then it cannot be a single factor because other factors also come into play. For example, skin lotions, body temperature, other factors come into play. So, in order to repel mosquitoes, if there are some other remedies which are available, they can be used, but also taking cognizance of the fact that we cannot use only one way to prevent getting bitten by mosquitoes. We need a combination of a number of factors, a number of strategies to prevent a mosquito bites. Thank you. I hope it has answered.
Yeah, great. Newton, I'm aware of time, but what medications are recommended as prophylaxis for people visiting areas where malaria is endemic? Can you answer that?
Oh, yes. Prophylaxis are definitely needed for those who are visiting areas that are endemic with malaria, but also taking note that we need to use this prophylaxis together with other available options. For example, you need to just avoid getting bitten by these mosquitoes. You need to make sure that if you are indoors, the designs of the buildings, if you are out, if you are not, if you are in an area that is endemic with malaria, just avoiding having these mosquitoes getting into the houses. We also need to make sure that there are some strong malaria interventions like spraying happening in those areas. So it's actually a cocktail of interventions which need to be taken into account. But when you are visiting an area that is endemic with malaria, obviously prophylaxis are needed.
Perfect. And I'd like to thank Dr. Oosthuizen for the article on chlorine dioxide. I've saved it. I'm certainly aware of chlorine dioxide being able to be a broad antimicrobial. We also know that doxycycline will treat viral, bacterial, and parasitic infections. And I think we need to open our minds to that. Dr. Carvallo's got back to me. The cabin crew still won't let him use his phone. So I'd just like to play what he said. I mean, one of the things that we found very interesting when we were researching ivermectin was that it stops vectors. And we know that from Kenya, but we need to use higher doses. And I'll just play you what he's actually said. And he will do a presentation at the Victoria Falls Summit in July on this. But just tell me if you can hear this.
Well, you know that when I'm on the plane, I have to close the phone. But I can tell you, I'm going to send you the effect of ivermectin on malaria, and also the effect of ivermectin on mosquitoes, which are somehow sterilized, but which see that their life expectancy is reduced to half. That's from 8 days to 4 days. That's quite important. It's amazing, to my opinion. And it's not exactly a matter of sterilization, but the ivermectin prevents female mosquito to transmit the virus to the larva. To its larva. So there's no vertical transmission of the virus when using ivermectin. And besides, ivermectin used topically on the water, which is the natural place for mosquitoes to grow, kills the larva more effectively than any other compound you may imagine, even far easier than the traditional compounds.
Okay, so I've spoken to Dr. Carvallo quite a lot. Spanish is his first language. So when he refers to virus, he's usually talking about an antimicrobial. He is an infectious disease consultant, but he's not a specialist in English. So I found that his input was quite useful, and I certainly know that a lot of people who were getting malaria regularly in Mozambique, it stopped when they were prophylaxing for other reasons. So I think that's an interesting comment, and it's open for controversy. So I think what we're seeing is a lot of people are seeing that these broad antimicrobial agents are not just restricted to being an antiparasitic or an antibiotic. The last question for the night comes, is it possible for a sickle cell anemia patient to contract malaria since they are considered immune due to the state of their hemoglobin? So Newton, sickle cell anemia, we don't see that much sickle cell anemia here. It's common in West Africa, obviously. But does anybody have experience with sickle cell patients? I've certainly never seen a sickle cell patient with malaria. And I saw quite a lot of them when I was working overseas, and they came back and specifically they didn't get malaria. but that's anecdotal. I haven't seen the evidence. So Newton, are you aware of sickle cell patients?
Thank you for that question. Sickle cell anemia patients, these people can contract malaria, from what I know, but the risk of developing complicated malaria is actually lower as compared to people who don't have sickle cell disease. I also have come across some literature which has indicated that they tend to have low parasite density, as well as people can develop malaria, but it's the severity which is lower as compared to the rest of the population. That's what I know. I think others on this platform might actually know better. Thank you.
So I think I think the important thing, I've got a direct message here, which is, can we please comment on blood volume? So why children are much more susceptible to malaria. And I think it's important to understand that once the parasite is injected, it starts to replicate. And we start seeing complications when the blood starts to sludge, which is when the parasite count is very high. It actually attaches to the CD147 receptor, and the blood starts to coagulate in the brain, in the kidneys, particularly in the glomerulus, and it also will coagulate in the capillaries of the fingers and toes. So if you leave malaria to day 5 in an adult, you may, on the next round of hemolysis, you may end up with a patient with a hemoglobin of 5 who basically strokes out and goes into renal failure and needs amputation of their digits. Now, if you are like most of us adults sitting with 5 liters plus of blood, then it's going to take a lot longer to get there. But if you're a 2-year-old child with 1 liter of blood, the speed at which you develop a parasite count of over 10% is much quicker. So we must be aware that in somebody who has a low blood volume based on their size and their surface to volume area, we're going to be dealing with cerebral malaria, renal failure, and loss of digits much earlier than in an adult. So your index of suspicion in a child and your speed of treatment needs to be a lot quicker. And I think that certainly my philosophy when I had my own children was I never took them into a malaria area until they were over 5 years old. And I'm not sure that was a rule of thumb from when I grew up. There's no real medical basis to that. But children under 5 were not invited on trips to the Zambezi Valley, as an example. They also tend to be prey for things like lions and leopards. So it's probably a good idea to keep them out of those kind of places. But I agree with that comment. Absolutely. The smaller the child is, if there's a fever of unknown origin, the higher your index of suspicion for malaria must be. And my feeling about sickle cell anemia, and Dr. Landman has asked this question, what harm can you do by giving coartem? And I've certainly never seen anybody die from coartem, but I've seen a lot of people die from malaria. So on a risk-benefit analysis, I would certainly favor giving medication if there's any doubt, particularly to a child. And I don't know if Ingrid, you want to comment on that, or Dr. Rapiti, you want to comment on that, or anybody who's seen a lot of malaria wants to comment. But I think the safe thing to do is take the blood, treat, and if you don't get confirmation, watch the clinical progression. So if the patient turns around on coartem or lumatem, you've got the diagnosis right. Ingrid, I don't know if you want to comment or anybody else wants to comment.
Jax, no, I agree with everything you've said.
Yeah. If in doubt, treat. I know that, you know, Newton and I had the conversation earlier that he's a researcher and I'm a clinician, and we're looking at the elephant from different ends. So I'm looking at the elephant and I'm seeing a trunk and I think it's a snake, and he's seeing the tail and he thinks that it's a rhino. But at the end of the day, we need to look at the elephant in the room from all angles, and we need to define our objective. And if our objective is good patient outcomes, then we must do a risk-benefit analysis and do what we can with what we've got. And that's my opinion. It's just an opinion. Dr. Lehman's also commented about low platelets. I haven't seen a malaria case that has a high platelet count. If you see low platelets in a pyrexia of unknown origin, suspect malaria, and have a very high index of suspicion, and a negative malaria test just means you need to repeat it on the next cycle. Dr. Rapiti, you're asking what anti-malarial prophylaxis you should use. I would say that it's regional. I have concerns with mefloquine because of the psychiatric effect in 10% of people. There's no point in going on a holiday and not being able to sleep and having nightmares with the prophylaxis. I know certainly in Zimbabwe, a lot of people use treatment as prevention. So the moment they get sick, they just treat. And I've certainly seen doxycycline used on a dose of 1 BD, used a week before and 4 weeks after going into malaria area. But I've also seen severe burns from photosensitivity from doxycycline. In this country, we use something called deltaprim, but I know there's resistance to it. So Newton, can you give us any specific anti-malarials that are recommended in Zim? I know that certain areas are considered as resistant. So Chiredzi has got resistant malaria, whereas Monopools doesn't. So I think you do need to go with local knowledge. And I don't know if anybody wants to comment on that. Newton or Ingrid, would you like to comment on what you use to prophylax?
I use Malinol, Tobaquan and Preguanol. If people are going, certainly if people are going to Mozambique. And believe it or not, some people like, for example, people who've grown up in this country and possibly have some level of immunity, going places like Kariba, then deltaprim is still an option. Because the problem with Malinol is it's expensive, Tobaquan, Preguanol. There is a generic, but it is expensive. So, yeah, and the doxycycline, I think, is a CDC recommendation. But it does have the issues of the photosensitivity in the sun. So I tend to use Malinol a lot.
Okay, thanks for that. I mean, local knowledge is really important. I've certainly, in the Lowveld with the resistant malaria, seen people break through on that. So, particularly if a patient has been on prophylaxis, you may delay the symptomatology. So generally, we say 10 days to 4 weeks in terms of the symptom presentation. But if a patient stops prophylaxis and then 2 weeks later develops a fever, also have a high index of suspicion that you've only partially suppressed them. I don't know if you'd agree with that, Ingrid.
Yeah, no, I would. I think high index of suspicion is always your first thing. And, you know, a couple of years ago, we had an outbreak of malaria in Harare, where there was a certain area in the north where there was a swamp and there was a whole lot of mosquitoes imported. And we've also had the odd freak case of people who picked up malaria that came from a sort of anopheles that came in on a bus and somewhere in Marlborough. So, you know, that they had not been out of Harare, but they'd been bitten by mosquitoes that came in on a bus from the Lowveld. So it doesn't matter where they come from. If their symptoms are not responding, always think of malaria without being paranoid.
No, I think it's good to be paranoid about malaria. I've certainly seen an extremely intelligent 5-year-old end up with a special needs class. She got it in Djibouti, and they misdiagnosed it, and she was treated with antibiotics, and she was flown to South Africa with a hemoglobin of 5, aged 5 years old, which in itself in a pressurized aircraft wasn't a good idea. But she very, very nearly died. She was in cardiac failure on arrival because people assumed that she was from a non-malaria area. So the safe thing to do is, if in doubt, do a therapeutic trial, which you and I both know was commonplace in the 1980s and the 1990s. Rumbidzai, you've asked, talking about resistance being mentioned, just to seek clarity. Is there resistance to coartem? My understanding is that there is. Lumatem is basically 4 times the dose of coartem, and it's certainly recommended for malaria in the Chiredzi area if you speak to the doctors at the Colin Saunders Hospital. So, yes, coartem resistance is possible. I think one of the things Newton mentioned is that a lot of people will stop treatment when they feel better. So it's very, very important to educate patients to complete a course. Obi, you've said 2 hands are up. I'm struggling to see them. I'm clicking on them, but nothing's happening. But that might be my technological incompetence. So can you just tell me whose hands are up and ask them to unmute their mics?
Okay, Dr. Westhuizen, I think you were up first. If you can dive in, please.
Yeah, Jackie, thank you. Did you refer to the file I sent you last week about CDS or today?
To the file you've just put on the chat.
Oh yeah, so I don't know if everyone saw it, but that's from the book of Andreas Kalker about the CDS and malaria. I'm very skeptical about allopathic medicines and GMO mosquitoes and all that since the COVID story. But I want to ask Dr. Newton, have you got any knowledge about CDS? It's in the protocol that I attached. I hope everyone could read it. I've treated a guy some 3 weeks ago, came from Mozambique. He had positive malaria, and I gave him CDS, but he wasn't quite better the next day and added doxycycline, and within a day he was fine, everything. And that's now 4 weeks ago. So it worked, I don't know, fully with me, but have you got any experience?
Thank you very much. Unfortunately, I don't have some experience with this. Maybe something that might need some further inquiry. Thank you.
Dr. Westhuizen, one of the things that has been very useful is Mr. Handireketi is the Deputy Director of the National Institute of Health Research. So we want to raise things that would potentially go into research projects. So I'll certainly take that protocol. And I think, we need research coming out of Africa rather than being told how to manage malaria by people who've often never seen it, and who don't have a healthy respect for it. So great comment. The other hand that's up, I'm clicking on the hand, but it's not doing anything. So Obi, who's the other hand?
Mr. Dr. Nyasha Chigariro.
Dr. Nyasha, please unmute your mic.
All right, thank you very much, and good evening, everyone. Thank you, Mr. Newton. Thank you, Dr. Jack, for this presentation. So mine is a contribution together with a question to Mr. Newton. So I think the gold standard for the diagnosis of malaria is microscopy, both on a thick and a thin slide. It's very unfortunate that we come from a very resource-limited setting, but otherwise the slide should be reported by a specialist hematologist or a specialist microbiologist. And if they don't see malaria parasites on a slide, they must spend at least 1 hour on that slide, and the second hematologist must also review that same slide to confirm that it is a negative, but very difficult to implement in our resource-limited setting in Zimbabwe. And I think the question to Mr. Newton, you touched a bit on sporozoids. I think that's the first time that I've heard of this. Would you like to elaborate a little bit more on that, especially telling us where it comes in? Because I think my understanding is that we do have the RDTs, which can either be falciparum specific or non-falciparum specific, and the microscopy is standard test. So where does the sporozoid come in? Thank you.
Okay, thank you very much for the question. So sporozoid is a stage-specific protein on the outer surface of the sporozoid. So this protein is what is tested for in the laboratory to determine if a mosquito is infective or not. So this information is, so when you catch mosquitoes and you want to see if the mosquitoes are infected, they carry the sporozoids. You can actually test for that particular protein as a marker for infection of the mosquito by the parasite. So this is where the sporozoid comes in. Okay, thank you.
Thank you, everyone. I'm aware that we are now half an hour over time, and the Namibians are giving me a hard time because we run on German time. So I think it would be a good idea to do another session on malaria. I think that we've become too focused on other illnesses, and we've let malaria and TB and leprosy and those other diseases get out of control. So I'd like to end the session there. Pride, can we stop recording?